What is life? Is this plant alive? Is the pot that it's in living? What about this turtle, this red slider? Is that living? How do we know? How do we decide what is living or not? Let's consider a few other objects here to try to see if we can differentiate between what may be alive and maybe what's not alive. For example, we've got a toy car and a wooden block. Now if I look at these three items and I take the turtle out of the equation, how do I know if one of these is living, or if maybe they're all living, or if maybe they're all non-living items? What I need to develop is a heuristic. I need some type of mechanism for identifying whether or not these items are living or non-living. And I care about this only because I'm a human, and as humans we tend to categorize objects. We look for patterns. So let's first ask ourselves a couple of questions. For example, is this toy car organized? It's got 4 wheels. It's got a structure that those wheels are attached to. It appears to be organized. In fact, it even moves in an organized fashion. Same thing with this block. It looks organized. It's compressed. It's got a nice rectangular shape to it. My plant is obviously organized. It's even organized in terms of the pot that it's in. What about something like homeostasis, a biologic term that means to maintain internal systems? Do these things do that? Well, my wood block doesn't do homeostasis, and my toy car doesn't do homeostasis, but my plant sure does. What about responding to stimuli? Right now my plant is in the sun. It's fully bathed in sunlight. And although we can't see it, on the leaves of the plant there are molecular reactions happening to help it photosynthesize in the sun. Unfortunately, while my car will respond a little bit to external stimuli, it doesn't respond in the biologic sense, nor would the wood. What about things like growth, reproduction, evolution? Again, I come back to the plant and I see that all of those characteristics could be true. At one time they may have been true for the wood block, which is constructed out of living materials, or what was once a living material. But now really it's only the plant that fits this model. And if I even ask one more question, is the plant made of a cell? Something like a parent cell that could then grow and give rise to 2 new daughter cells. We find again that the only object here that could do that trick currently would be the plant. Again, some of these were materials that at one time may have been called living, and maybe we can even now differentiate and say this is the living and this is the non-living, and this is the once lived and this is the never lived. But if we really want to know what is life, we got to go back to this basic unit, the cell. So how did we actually come up with this idea of cells? Well, it took a couple of different collaborators to help us out with this idea. In fact, before anybody came on the scene, the first thing was this object here, cork. It turns out in the 1700s, early on, there was a fellow named Robert Hooke that really was interested in looking at cork. Not cork like you'd get from a wine bottle like this, but cork from the cork bark tree. And he was the first person, the first scientist to actually look at cells under the microscope and coin the term cell. And then he was also joined in collaboration by another person named Van Leeuwenhoek. Now, fast forward about 150 years, and we come to two very influential people. We've got Matthias Schleiden and Theodor Schwann. And both of these individuals actually worked together to form the first part of what we now know as cell theory. Cell theory helps us understand, essentially, initially, the first part, that all life comes from living cells. Now, that was a great start to this idea, but we need to continue on to see where this also goes. It turns out that Schleiden and Schwann still thought cells came from a spontaneous event. And now we know, thanks to Rimac, initially, that cells actually come from other living cells. Now, that idea was actually taken by Virchow and used, and he's the one that earns credit currently. But it was Rimac first, looking at chick embryos and frog spawn, that figured this whole puzzle out. So all of the people that were involved with cell theory, Schleiden and Schwann and Rimac and Virchow, all helped us to start to understand what was actually inside the cell, those component parts called cells, and then even deeper into the cell, things like our DNA. Now, our DNA is highly wrapped into these little structures called chromosomes, but pre-chromosome, it tends to look more like this. It's got a spiraling structure with different types of bases, A's, T's, C's, and G's, all lined up in what we call a double helix. Now, this was an incredibly important discovery, and this actually helped push science in terms of cell biology. First, we had people like Leeuwenhoek down here helping us see rudimentary things under the microscope. Then we started to see actual cells, both eukaryotic and prokaryotic, and then structures of cells over time. And then we got into things in our more modern area, things like the structure of DNA, looking at that very carefully, the structure of RNA, another type of nucleic acid. And even as recently as the 1990s, we've had full eukaryotic copies made of items like sheep. So it turns out that cell theory was actually an incredibly important discovery that helped guide the rest of cell biology as we know it. Now we're in the modern era, and we can actually rapidly sequence things like DNA. So all of these things were incredibly important to what we now know as cell biology. Now, if we look a little bit bigger, if we take a slightly larger view and we step outside of the realm of something really tiny like DNA, we have to ask ourselves, well, where do we find structures like that? Now, there are two main types of cells. There's what we call a eukaryotic cell. This is an example of one of those. This is a plant cell. And this would be another example. This is an animal cell. Both of these are very similar. They're both eukaryotic. This would be a good example of something like a prokaryotic cell, like bacteria. Now, these differ in terms of their component parts and their structures for the most part. For example, prokaryotes still have DNA. It's just wrapped a little differently and shaped more in a circular shape than what we would typically see, like a linear shape for eukaryotes. Also notice there are far fewer structures in this cell than you would find in a typical eukaryotic cell. You do have DNA, you do have ribosomes, you do have a cytoplasm, and you even have a plasma membrane that encases the entire cell along with a cell wall. But then notice that in the eukaryotic cell, you've got a whole lot more going on. You've got these things called organelles, little mini organs surrounded by membranes. So for example, if we were to look here at this animal cell, we've got centrally located the nucleus, and inside there's the nucleolus. That's where the ribosomes of the cell are made for protein production. The nucleus, notice, is a double membrane-bound structure that has pores coming out of it to have the actual genetic material in the form of RNA leave the nucleus to come out into the cytoplasm, and that's for information sharing. You've also got these interesting structures like the rough endoplasmic reticulum, which is dotted with little ribosomes to make proteins. That's coincident with the nucleus. And the smooth endoplasmic reticulum, no ribosomes here. The structure makes lipids and other hormones for the cell. And then you've even got these interesting structures called the Golgi apparatus. And there are a couple here in the plant cell as well. We've got one up here, for example. And these have little vesicles that bud off of them. This is where a lot of packaging and sorting and tagging for the cell happens. Now, near some of these other structures, you've got smaller components in the cell, which are still considered organelles. Things like here, this is the lysosome. Lysosomes, if you look at an open one here, are the garbage trucks of the cell. They do a lot of detoxification. And they're much smaller than organelles like the mitochondria, which are energy producers for the cell. Now, mitochondria are related to another type of organelle like the chloroplast. And these are specific to the plant cell. These actually capture light energy from the sun and help make sugars. Notice plants have mitochondria too to then break down those sugars. And then within the plant cell, a big structure that animal cells don't have is the central vacuole, which we think may actually serve a similar function to the lysosome. So in a fully functioning cell, it's not a static environment like you see here. It's actually very dynamic. And those are healthy functioning cells, ones that have low entropy, low disorder. However, sometimes bad things can happen to our cells. In terms of a prokaryote, something that could actually serve to attack a prokaryotic cell would be something like a bacteriophage. These interesting little structures are viruses. They're not living structures because they don't maintain all the characteristics of life we previously covered. But they do have biologic components to them, and they actually follow things like the lytic cycle. They actually lyse open the cell and release copies so that they can be then replicated by the organism overall. Another nefarious invader would be something like a retrovirus. This is more apt to invade a eukaryotic cell. And this is even more insidious in that it actually comes into the cell and it corrupts the DNA of the host cell, and then it uses that as a factory to make copies. More recently, we've started to become concerned about things like prions. If this pipe cleaner actually represented something like a prion is a type of protein. Proteins are biologic structures that are typically folded and perform lots of functions for the cell. If this were the normal type of protein, and this were the abnormal form, or the prion we would call it, these are pretty insidious in that they can just simply become close enough to the normal shaped protein, and they can actually cause the normal shaped protein to fold into the same conformation. And that can be very deadly for the cell in that it ends up creating plaques and openings and can possibly destroy neurologic tissue. So it's actually quite complicated to become something that's living and to maintain these structures over time. It takes a lot of energy put into the system. But life is one of those things that just keeps going no matter how many things get thrown at it. So it's an interesting thing to consider how life works, how we got here, and how our knowledge is going to keep growing.